Good evening. Welcome to our new intensive revision series. So this is a 45-day series where we'll be focusing on rapid revision of all the important topics. All the volatile quick facts and high yield topics will be quickly covered during this series so that you're able to benefit in your university exams and the approaching NEET PG exams and FMG exams as well. So in today's class, we'll be talking about vitamins. So vitamins are a very important topic, very frequently asked, your fat solubles and water soluble vitamins. So we'll be discussing their chemistry, sources, absorption, daily requirement, biological functions, and deficiency manifestations. So all of this is like really important, and you can see the number of times it has been asked in 2009, in 2003, 2006. So multiple times they've been asked. Then you get these in long essay types. Then you also get questions like your chemistry, sources, metabolic functions, again very important topic, and then the deficiency manifestations, again associated with all the vitamins. Here in this question, you can see for vitamin C, it was asked twice in 2005 and in 1999. Then you have short essay types questions as well, where you'd asked about like name a provitamin form of vitamin A. So these MCQ types or short questions where you can be asked to write short notes on the biological functions or their sources. Your vitamin A is really very important. Then you can see the number of times this question has been asked, like in 2009, 2010, 2011, 12, 18, 20, 2009, and 2002. So it has been asked like almost every year consecutively. So what your vitamin A is like really important. Then you have vitamin D, its functions and causes. Again, rickets is very important there. Then your coenzyme roles of various vitamins. Then in short answers, you have Figlu excretion test, folic acid role in DNA synthesis, then causes of scurvy, beriberi, rickets, and then you have your daily requirements of thiamine, niacin, pyridoxine have been asked multiple times in different years. So we are going to cover the entire of vitamins in this class today. Then introducing you to Auricle. Why do you have to choose Auricle above all? So we have very unique features here in Auricle. We provide you with recorded lectures, live classes, practical sessions, which is something very unique about Auricle. You'll not find this in any other institute. So we also have exam modules and hard copy book sets available. So in the recorded classes, you have the facility of flexible learning. You can view the lectures as per your convenience anytime, anywhere. This is going to be very convenient for your revision. Your lectures have been curated by expert faculty members. Then at live classes, you have the facility of interactive sessions. You can ask your questions then and there, and hopefully they'll be resolved. Then we have express exam modules, which covers the complete syllabus in 20 hours. Then you'll also be told about the answer writing skills in the exams by the expert educators. Then you also have one-to-one doubt clearing sessions from Auricle, which is going to be really personalized and student centric approach, followed by practical sessions. So you're going to have a hand-on experience through the expert guidance of the faculty. Then question bank, again very important. So everything that you've learned, you can assess your knowledge on the basis of the question banks. We have questions banks at 3 different levels. Depending upon your level, you can choose the one you want to practice. Then we also have recently launched a hard copy notes. So these notes have been specially curated, very precise, very concise, but at the same time not excluding any information. Okay, everything that is relevant is there in the notes, very colorful. So you can buy our notes if you want to prepare for your approaching exams. Then at Auricle, we have 3 different subscription plans. The first one, the basic, which comes at 20,000, followed by the standard plan, which is for 35,000, and then the premium plan, which is for 60,000. In the premium plan, you get one-to-one personalized guidance and notes as well. Then apart from that, you can get additional 20% off from the promo code displayed at the bottom of the screen. Okay, so let's begin with our class. Already our class has been delayed today because of some technical issue. So without wasting much time, Let's start. So before introducing you to the individual vitamins, let's take an overall introduction of vitamins. That what are vitamins? So your vitamins, they're organic compounds present in very small, very minute quantity in various food substances in our body. They are needed for growth and maintenance of the body. So they are dietary essential. Okay, that is, they're not synthesized by the body. You have to consume your vitamins through diet, with the exception of certain endogenously synthesized vitamins. So, like we all know, exceptions are always there in biology. So, as a general rule, your vitamins are dietary essential, but we do have endogenously synthesized vitamins in the body. So, they are organic compounds needed in very small, minute quantities, various food substances that are going to help you in the normal growth and maintenance of the body. Okay. So, what are the endogenously synthesized vitamins? So, in the endogenously synthesized vitamins, you have two categories, that is by the body and in the body. By the body means that your body itself is going to synthesize that vitamin. For example, your niacin, which is derived from tryptophan, or your vitamin D, which is synthesized from 7 dehydrocholesterol by exposure to UVB rays in the 290 to 315 nanometer in the sun. So, we're going to, when we study vitamin D, we're going to see how it is synthesized. But for now, you know that you vitamin D, which is synthesized in the skin when exposure to the UVB rays in 290 to 315 nanometer, and then you have niacin, which is derived from the amino acid tryptophan. So these are the two endogenously synthesized vitamins which are being synthesized by your own body. Then you have some vitamins which are synthesized in the body, that is somewhere inside the body they are being synthesized, but not by the body, they're being synthesized by somebody else. And what is that somebody else? That is your intestinal bacterial flora. So by the bacterial flora in the intestine, you have these vitamins being synthesized, that is your vitamin K, biotin, pantothenic acid. Okay, so now if a question comes, for example in an MCQ, you get something like, which of the following is an endogenously synthesized vitamin? And your options are, you have niacin or you have vitamin D followed by vitamin K, biotin, pantothenic acid. So in that case, the preference is going to be given to the vitamin that is synthesized by the body. That is, you're going to prefer niacin and vitamin D over vitamin K, biotin, and pantothenic acid. Okay, so always remember the preference is given to the vitamins that are synthesized by the body. If a question comes something like, which of the following is an endogenously synthesized vitamin, don't get confused that vitamin K is also there and vitamin D is also there, so both of them are being synthesized in the body to let me mark vitamin K. No, always remember your vitamin K, biotin, and pantothenic acid, they are being synthesized in the body, but it is done by the bacterial flora of the intestine. Okay, now moving ahead to the classification of vitamins. So your vitamins, they have been broadly classified into fat soluble, which includes vitamin A, D, E, and K, and water soluble vitamins, that is vitamin B complex and vitamin C. So let's talk about the variations or differences in fat soluble and water soluble vitamins. So your fat soluble vitamin, since it is a fat soluble vitamin, it is going to be absorbed through chylomicrons. They are stored in liver and adipose tissue. They are not excreted. Of course, when they're being stored, I am saying they're being stored. That means they're not being excreted. And since they are not being excreted and stored, they have toxicity and they have varied functions, except for your vitamin K. That is the only fat soluble vitamin which is having a coenzyme role. So your vitamin K has a coenzyme role. Otherwise, your B complexes, they are usually having coenzyme function. Water soluble vitamins, they do not need chylomicrons because it can be directly absorbed from the intestine. They can be directly absorbed from the intestine. Then they are not stored. Excretion, they are excreted in the urine. They do not have toxicity as such except for vitamin B6 and niacin, and they have coenzyme function. Now let's begin with vitamin A, very important. So your vitamin A, it includes retinoids and carotenoids. The active retinoids is your active vitamin A, and you get it from animal source. So basically from animal sources, you're getting retinol, okay? Compounds which are chemically related to retinol, they are your retinol and retinoic acid. So strictly speaking, your vitamin A in the strictest sense is your retinol, okay? And other biologically related compounds to retinol, that is your retinol and retinoic acid, they are also considered under retinoids, okay? But in the strictest sense, what you're getting from animal sources, it's your retinol. So your vitamin A in strictest sense is retinol. Okay. Now moving on to carotenoids. Your carotenoids, they are pro-vitamin A. That is, it can be converted to active vitamin A. Can be converted to active vitamin A. Okay. You get carotenoids from your plant sources. Most prevalent carotenoid is your beta carotene. Okay, so your beta carotene is the precursor for your retinol. Now, the richest source of beta carotene is carrot. There are about 600 carotenoids, and some examples being beta carotene, which is an antioxidant. Lutein, zeaxanthin, both of which are used in the treatment of macular degeneration. In treatment of macular degeneration. And then you have lycopene, which is used in the treatment of prostate cancer. Okay, so we've spoken about vitamin A, that your vitamin A, it has two forms. That is, it is including retinoids and your carotenoids. Your retinoids, they are the active vitamin A, which you are getting from animal sources. And vitamin A in the strictest sense is your retinol, okay? Apart from that, you have other retinoids in different forms. That is your retinol, which is having aldehyde as a functional group, your retinoic acid, which is having carboxylic acid, and then of course your retinol, which is having alcohol as the functional group. So your vitamin A is often used as a collective term for several related biologically active molecules. That is your retinol, and retinoic acid. So a collecting term for all these biologically active molecules which are related is your vitamin A. Then the other form that you have is carotenoids. But carotenoids, they're existing as precursor molecules, okay? They are your pro-vitamin A, which can be converted to its active vitamin A form, and you're getting them from plant sources. The most prevalent carotenoid is your beta carotene. So your beta carotene is a precursor molecule. We will see how this beta carotene can be, by the action of an enzyme dioxygenase in the intestine, cleaved to give your retinaldehyde or retinol, which then gives retinol. Okay. Then the richest source of beta carotene is carrot. There are about 600 different carotenoids, and to name a few, your beta carotene, lutein, xanthine, and lycopene. Now let's talk about the structure of vitamin A. So your vitamin A, it has beta ion ring, single, that is one, and isoprenoid chain. So what is iso? Your fat soluble vitamins, remember, they are actually isoprenoid compounds formed because of condensation of isoprene unit. What is an isoprene unit? It is a 5 carbon unit, and it looks like this. Okay, so this is your isoprene unit. Okay, so you can see here in the structure of vitamin A, it is having, this is your beta ion ring, so it is having a single beta ion ring, and you can see these isoprene units. Okay, so they have multiple isoprene units, that is isoprenoid units. Okay, so all your fat soluble vitamins, they have isoprenoid chain. So that is the structure of vitamin A and your beta carotene, but it is having two beta ion rings. You can see one here and one here. And this is the site of cleavage that I was talking about. So your beta carotene, it is having two beta ion rings. It is going to be cleaved to give retinal and retinol. Okay, so this is the structure of your vitamin A. This is the structure of all trans retinal, and this is the structure of beta carotene. Remember, all trans retinal is having a single beta ion ring, and your beta carotene, it is having two beta ion rings. Okay, now your different forms of vitamin A, they can be interconverted. Okay, that is your retinol. It can be oxidized to retinol. We already know that alcohols, they're oxidized to aldehyde groups. Okay, so your retinol and retinol, they are readily interconvertible. Okay, so your retinol, it can form retinol, and your retinol, it can form retinol. And this retinol, on further oxidation, it gives retinoic acid. But this retinoic acid, this cannot be reduced in the body to give either retinol or retinol. Okay, so your retinol, that is alcohol, it can be oxidized into retinol, and retinol can also be reconverted back into retinol. So they, retinol and retinol, they're interconvertible forms, but retinol on oxidation forms retinoic acid, and this retinoic acid, it cannot be reduced in the body to give either retinol or retinol. Okay, therefore it regulates gene expression. So your retinoic acid, it is involved in gene regulation, whereas your retinol and retinol, they are involved in Walsh visual cycle, because they are interconvertible forms. Later, when we're talking about the functions, we'll see how. Okay, now let's talk about vitamin A metabolism. So talking about its absorption, transport, and storage. So instead of this diagram, I'm going to make a hand diagram so it will be easier for you to understand. Okay. Now, let's consider that this is the intestinal lumen, and this is the intestinal cell. Okay. Then from the diet, you're getting vitamin A. Okay, so through the diet that you've consumed, you have vitamin A. Now, from the plant sources, we've already studied that the most common, mainly you're getting carotenoids, and the most common in carotenoids is your beta carotene. So the from plant sources, you get carotenoids, and the most prevalent carotenoid is your beta carotene. So you get beta carotene here in the intestinal lumen. And from animal sources, you're getting your retinol is found in animal tissues as retinol esters. Okay, so from animal sources, you're getting retinol esters. So from animal sources, you're getting retinol esters, because retinol in animal tissues is found ester to your fatty acids. Okay, so you get retinol esters. Now what happens in the intestinal lumen? Your beta carotene, as it is absorbed and it enters the intestinal cell. Here in the intestinal cell, what is happening? It is going to be acted upon by an enzyme, dioxygenase. And what is, we know that beta carotene, it has two ion rings, and then in the center of it, the cleavage was going to happen. So on cleavage, what is going to happen? Your retinol is formed. Now your retinol esters that were coming from the animal sources, what happens to that? It is hydrolyzed, the fatty acid is removed, and you end up with retinol. So your retinol, it was into an ester bond with the fatty acids. Okay, it was esterified with fatty acids, and the most common fatty acid in retinol ester is your palmitic acid. So your retinol ester, it was having retinol plus fatty acid, and the most common fatty acid is palmitic acid in this. So the fatty acid is removed, and now this retinol is absorbed and it enters the intestinal cell. Now, in the intestinal cell, what is happening? The retinol that you're getting from beta carotene, that combines with fatty acid, and this retinol that you're getting from the animal sources, okay, this also combines with fatty acid, and what is formed is retinol esters. Okay, so again, retinol esters are formed in the intestinal cell, and these retinol esters, they are carried via chylomicrons. These chylomicrons, they're having the ligand ApoE. Okay, so this is how your vitamin A is absorbed, okay, and carried via chylomicrons. Now let's see what the notes have to say. So the retinol esters from animal sources. Okay, we know that we're getting retinol esters from animal sources and beta carotene from plant sources. They're absorbed from the intestinal lumen into the intestinal cell. Okay, then they are transported via chylomicrons and reach the storage organ, which is liver. Okay, so your chylomicrons, if you remember when we were doing the metabolism of LDL, HDL, chylomicrons and all. So we studied that your chylomicrons, they were involved in carrying exogenous, they carry exogenous, that is dietary tags. Okay, that is whatever exogenous or dietary tag that you're having, they are carried in chylomicrons from the intestine to the liver. Therefore, your vitamin A, which is again a fat soluble vitamin, it is carried via chylomicrons, okay, from the intestine to the liver. And your chylomicrons are having the ligand ApoE, and the receptor for ApoE is present on the liver. So your liver is having receptor for ApoE, and the ApoE ligand is present on chylomicrons. Okay. Now, all fat-soluble vitamins, they are carried to storage organs via chylomicrons, okay? Because they're all fat-soluble vitamins, therefore, they cannot be directly absorbed. They are carried through chylomicrons. Now, retinal esters are stored inside the liver in perisinusoidal cells, also known as ETO cells. When the body requires vitamin A, it is transported in the blood to the target organs with the help of two transport proteins forming a ternary complex, that is tri-molecular complex. So after being stored in the liver in the ETO cells, what happens when your vitamin A is required by target organs? Then with the help of two transport proteins. Okay, these are the two transport proteins. What are they? Retinol binding proteins and your transthyretin. And this is your vitamin A here. So this ternary complex, tri-molecular complex is formed bit of the vitamin A and the two transport proteins. Okay, and then from your liver, it is transported to the target organs. What are your target organs? Where your vitamin A is required, like in the eyes, in the skin. So organs where the action of vitamin A is required, those are your target organs. Okay. And why transport proteins are needed? Because again, your vitamin A is not soluble in blood. Okay, and this transthyretin, trans is for transporter. Rectin because it is transporting your retinol. And also remember thigh because this transporter is also involved in the transport of your thyroid hormones. Okay, so this is also transporter for thyroid hormone. Remember, transthyretin. Okay. So we've studied how your vitamin A, it was absorbed from the intestinal lumen and then carried via chylomicrons, reached liver, stored in the liver, and then when it was needed by target organs, Via two transport proteins forming a tri-molecular complex, it reaches the target organs. Now, let's have a look at the functions of vitamin A. So, the most important function is vision. Form of vitamin A needed for vision is your 11-cis retinol. Then skin and mucosa, your vitamin A is needed there for maintenance of epithelium. It is also needed in reproduction, in regulation of gene expression. The form needed is retinoic acid. Why retinoic acid? Because your retinoic acid, it cannot be reduced back into retinol or retinol. Then it is needed for growth and development, for tissue differentiation. It is also acting as an antioxidant, the most important antioxidant being your beta carotene. Then beta carotene also has photoprotective properties. Therefore, your beta carotene can be applied in cutaneous photosensitivity. Cutaneous photosensitivity. Okay. Now let's have a look at the Walsh visual cycle. So your 11-cis retinaldehyde, it is going to combine with a protein known as opsin to form rhodopsin. And this rhodopsin is known as visual purple. Now what's Walsh visual cycle? We'll see. But before that, for dim light vision, I'm sure you all know, you need a protein known as rhodopsin, which is present in the rods. And for colored vision, your cones are required. Okay, now let's have a look how your Walsh visual cycle is working. So this is what is happening inside the retina. Okay, your all trans retinol is changing to 11-cis retinol. Then this 11-cis retinol changes to 11-cis retinaldehyde. Now this 11-cis retinaldehyde is going to combine with a protein which is known as opsin. Okay, so your 11-cis retinaldehyde combines with the lysine residue present in a protein opsin to form what is known as rhodopsin or visual purple. Now, what happens when light falls on this visual purple? A series of photoisomerization reactions takes place. You can see when light is falling on this visual purple, a series of isomerization reactions. A series of photoisomerization reactions occur. Okay, so these are taking a fraction of a second. Okay, and as a result of which your rhodopsin is converted into different isomeric forms like photorhodopsin, bathorodopsin, lumirhodopsin, metarhodopsin 1, 2, and 3. Now, this metarhodopsin 2 that is formed, this is an important isomer because it ignites signal cascade pathway. It ignites signal cascade pathway. How? By activating a G protein known as transducin. I'll just discuss how. Then, for now, just know what happens when light falls on rhodopsin. A series of photoisomerization reaction takes place. Different isomeric forms of rhodopsin are formed, and out of which the form metarhodopsin 2 is important because it is able to ignite a cascade signal cascade pathway. Then what happens? Your metarhodopsin 2 changes into metarhodopsin 3, and then from metarhodopsin 3 your opsin is released. Okay, so your opsin is released. You can see here opsin is released and all trans retinaldehyde. So your metarhodopsin 2 releases opsin, and what is left is all trans retinaldehyde. This all trans retinaldehyde is again going to convert to all trans retinol. Here it is going to convert to all trans retinol, and again a new cycle is going to start. So you see this is happening in a cyclic manner, which is known as Walsh visual cycle. Your all trans retinaldehyde is again going to convert to all trans retinol. Then all trans retinol will again form 11-cis retinol, then 11-cis retinaldehyde, which is going to combine with the lysine residue of opsin, and again rhodopsin is going to be formed. Then light is going to fall, series of photoisomerization reaction, and this is what is known as the Walsh visual cycle. So this is how your vitamin A is involved in vision. Now let's have a look how your metarhodopsin 2 is actually able to ignite a signal cascade pathway. So what happens is your metarhodopsin 2, it activates a G protein coupled receptor known as transducin. Now this transducin is going to activate an enzyme known as phosphodiesterases. And phosphodiesterases, when they're active, they convert cyclic GMP to 5' GMP, and this 5' GMP, it is going to act on sodium channels and close it. And as a result of closing of sodium channels, what is going to happen? Hyperpolarization is going to happen. Hyperpolarization. And this generates a nerve impulse, and you're able to see. Okay, if you have a look at this diagram, you see what is going to happen. Your metarhodopsin is going to activate transducin. Transducin is a G protein coupled receptor, and in its inactive form, it is bound to GDP. When it is activated, it is bound to GTP. Now, this active transducin, it is going to activate phosphodiesterases, and phosphodiesterases, when they're active, they convert cyclic GMP into 5' GMP. And what are 5' GMP doing? They are closing your sodium channels. Okay, and as a result of closing of sodium channels, expression. Now, retinoic acid binds to nuclear receptor and activates it. Active nuclear receptor then binds to response element of gene and regulate gene expression. Retinoic acids acts via nuclear receptor just like steroid hormones. So what is happening is your retinoic acid, it is going to bind to nuclear receptors. What are nuclear receptors? You have two types of receptors. One that are those are present in the plasma membrane, and the other those are present in the cytoplasm. Okay, so they are going to bind to nuclear receptors. We're going to study this in detail when we talk about signal transduction. Okay, and then they are going to bind to response element. Okay, this is your response element on the DNA and regulate gene expression. Okay, so what form of vitamin A is involved in gene expression? That is your retinoic acid. Retinoic acid binds to nuclear receptor, which then binds to response element of the DNA and regulate gene expression. Now let's talk about two types of receptors of vitamin A, that is RAR and RXR. Okay, so RAR is retinoic acid receptor. Form of retinoic acid that acts as ligand for RAR is your all trans retinoic acid. So all trans retinoic acid, for that the receptor is RAR. Active RAR binds to response element and regulate gene expression. Now you have another receptor, that is RXR, retinoic acid X receptor. The form of retinoic acid that acts at ligands is 11-cis retinoic acid. So 11-cis retinoic acid is going to work via RXR receptor. But what is the difference? Your RXR is going to dimerize with active vitamin D receptor, and it can also bind to thyroid hormone receptors. Now this heterodimer bind to the gene and regulate gene expression. So your RXR is going to bind to the response element on the DNA as a heterodimer, and your RAR is going to bind as a monomer. This vitamin D deficiency can also cause vitamin A deficiency, or thyroid hormone deficiency can also lead to impairment of vitamin A functions. Therefore, active RXR can bind to gene only in heterodimer form. If I explain this to you with the help of a diagram, I think it will be more clear. See, These are your two receptors. Vitamin A, I said it has two types of receptors, RAR and RXR. Now for RAR, the ligand is your all trans retinoic acid. You can remember this RAR, it has A in its name and all trans A. So you can remember it that way. And for your RXR, the form of retinoic acid is your 9-cis retinoic acid. Is the ligand for RXR. Now what happens? Your all trans retinoic acid is going to bind to RAR, and then this activated RAR is going to bind to the response element on the DNA. This is the response element on the DNA. And bring about regulation in gene expression. But RXR is going to work differently. It is first going to form a heterodimer either with active vitamin D or thyroid hormone receptor. So what happens? Your 9-cis retinoic acid binds to RXR. Now what is going to happen is that this RXR binds to vitamin D receptor. So this is RXR, and this is going to bind to vitamin D receptor. Okay, so this is your vitamin D and this is your vitamin D receptor, or either thyroid hormone receptor. Now you see a heterodimer response. Now this heterodimer is going to bind to response element and then bring about regulation gene expression. So that is how your two different receptors work. Now let's talk about the deficiency of vitamin A. Again, important. Firstly, the deficiency manifestation seen in the eyes. So that is loss of vision to green light. So this is the earliest sign and not symptom. This is the earliest sign and not symptom. Why? Because the patient will not be able to recognize this. Then nyctalopia, that is night blindness. Then dryness in the conjunctiva and cornea, that is dry eyes, that is erophthalmia. So your earliest sign is your loss of vision to green light, and nyctalopia is your earliest symptom. Okay. Then keratitis, that is inflammation of the cornea. There will be photophobia in this case. Then bitospores, that is raised area in conjunctiva. Then you have corneal ulcers known as keratomalacia. So your vitamin A is the most common vitamin deficiency and also the most common cause of preventable blindness. Then manifestations on the skin, follicular hyperkeratosis, also known as papillary dermatitis or toad skin. Then maintenance of epithelium needs vitamin A. So your vitamin A is very necessary for the maintenance of the epithelium. Then talking about the toxicity, usually it is rare, but it is seen in Arctic explorers who basically depend on polar bear for food. So the liver of the polar bear is going to store vitamin A, since liver is the storage organ of vitamin A. So they will end up in having excess vitamin A leading to toxicity. So the toxicity manifestations, they are your headache, vomiting, nausea, hip fractures. Why hip fractures? Because vitamin A is going to increase your osteoclastic activity. Okay, and as a result of which there will be increased resorption on the bone and increased fracture susceptibility. Then talking about the therapeutic applications, it is photoprotective, therefore used for cutaneous photosensitivity. What is your beta carotene is applied. All trans retinoic acid, it is used for mild acne and used to prevent skin sagging. Or your tretinoin, okay, and they are not teratogenic. Then your 11th is retinoic acid, that is your isotretinoin. Isotretinoin, it is used for cystic acne and childhood neuroblastoma. Now, this your isotretinoin or your 11-cis-retinoic acid, it is teratogenic. Okay, that is, it is going to interfere with fetal development. Therefore, it is not pregnancy and lactation safe. Then sources and RD of vitamin A, your retinoids. The animal sources are liver, egg, butter, cheese, milk, fish, meat. Carotenoids you're getting from plant sources like carrot, mangoes, and papaya. Talking about the RDA, in children it is 400 micrograms per day. In pregnancy it is raised, that is 800 micrograms per day. In lactation it is 950 micrograms per day. And for men and women, normally it is 600 micrograms per day. So we've spoken about vitamin A. Let's talk about vitamin D. Okay. So this is another fat soluble vitamin, that is your vitamin C. It is a group of sterols which have hormone like actions. So it is also known as sunshine vitamin. We'll discover very soon why. The compounds considered as vitamin D, they are your vitamin D2, that is ergocalciferol, that is coming from plant sources, and D3, that is cholecalciferol, coming from animal sources. So, firstly, talking about the sources of vitamin D. So, it is an endogenously synthesized vitamin. Your vitamin D, it is synthesized in the skin from 7-dehydrocholesterol. What is happening when UVB rays in 290 to 315 nanometers, they fall on skin. What happens? Photoisomerization reactions are going to take place. This is a two-step reaction, and your 7-dehydrocholesterol is going to be converted to cholecalciferol, that is D3, inside the skin. So this is how your cholecalciferol, vitamin D3, is formed in the skin from 7-dehydrocholesterol. Now, all diet is poor in vitamin D unless fortified. That is, unless vitamin D is added, majorly the diet is a poor source of vitamin D. Preformed vitamin D is a requirement only in individuals with limited exposure to sunlight. Okay, so if you have limited exposure to sunlight, then only you need to take vitamin D. Otherwise, it is being formed endogenously in your body, in the skin. Now, from the diet, which is a very minor source, because majorly it is synthesized in the skin. So you're getting ergocalciferol. Okay, that is a plant source from like ergots. And you're getting cholecalciferol from animal sources like fish. Okay, so otherwise, apart from fish, I said only the only source of vitamin D is fish. Otherwise, apart from that, all diet, unless it is fortified, it is a poor source of vitamin D. Okay, so from animal sources like fish, you're getting cholecalciferol, and from plant sources like plants like ergots, you're getting ergocalciferol. Your ergocalciferol is also converted to cholecalciferol. So finally, in the body, you are getting cholecalciferol through diet, and also from 7-dehydrocholesterol in the skin, your cholecalciferol is being synthesized. Okay, so ultimately in the body, you are having cholecalciferol either directly from plant sources. You're having cholecalciferol, or either you're having ergocalciferol, which is converted into cholecalciferol, or from 7-dehydrocholesterol, your cholecalciferol is being synthesized. So ultimately, you in the body, we're having cholecalciferol, that is calcidiol. It has one hydroxyl group. Okay, remember that. And also 7-dehydrocholesterol, it is an intermediate in cholesterol synthesis as well. Therefore, we say that vitamin D is a steroid. Okay, now let's talk about metabolism of vitamin D. So through diet, which is very less amount, a minor source, or through endogenous synthesis, which is the major source, we are having cholecalciferol inside our blood. Now, with the help of transport protein, vitamin D binding protein, that is, your cholecalciferol reaches liver. So in the blood, you have cholecalciferol. Now, through the help of transport protein, since it is a fat soluble vitamin, so you need transport proteins. What is happening? It is going to reach the liver. Now, in the liver, what is happening? There is an enzyme known as 25 hydroxylase. Okay. So this belongs to cytochrome P450. Now, this 25 hydroxylase, it is going to hydroxylate your cholecalciferol, which is also calciol, because it is having one hydroxyl group. Okay, so it is already having one hydroxyl group. It is known as calciol. Now what happens in the liver? There is an enzyme 25 hydroxylase. It is going to hydroxylate this cholecalciferol at the 25th position, and it becomes calcidiol. Why diol? Because now it has two hydroxyl group, one at the 25th position and one that it was previously having. So you have calcidiol, which is 25 hydroxy cholecalciferol. Now, this calcidiol from the liver is transported to the kidneys, where one more hydroxylation takes place by the enzyme alpha 1 hydroxylase. And this alpha 1 hydroxylate is favored by parathyroid hormone and considered as the rate limiting enzyme. Okay, so now in the kidneys, another hydroxylation at the first position is taking place, and your calcidiol now becomes calcitriol, because now you have three hydroxyls in its structure, one at the first position, one at the 25th position, and one that it was initially having. So you end up with calcitriol, which is 125 dihydroxy cholecalciferol, and this calcitriol is the biologically active form of vitamin D. So you have a look at this diagram. It'll be more clear. So you have cholecalciferol, okay, synthesized in the skin from 7 dehydrocholesterol. Okay, now this cholecalciferol, it is going to reach the liver. Now in the liver, you have this enzyme 25 hydroxylase, and cholecalciferol will undergo hydroxylation at the 25th position, and you end up with calcidiol. Now this calcidiol reaches the kidney. In the kidney, hydroxylation is taking place by alpha 1 hydroxylase, as you can see, and you end up with the biologically active form of vitamin D, that is calcitriol. And this alpha 1 hydroxylase in the kidney, it is activated by low phosphate and parathyroid hormone. Okay, and it is inactivated by calcitriol itself. Now, what happens when the body does not require vitamin D? It is converted to its inactive form by 24th hydroxylation. So, if the body is not requiring vitamin D, there's already enough levels. So, what is going to happen? Hydroxylation at the 24th position is going to take place to inactivate this active vitamin D. So, in the kidney, 24 hydroxylation forms calcitetrol. So, if this 24th hydroxylation is taking place in the kidney, it is known as calcitetrol, because you will have 4 hydroxyls now. And if it is taking place in the liver, with the 24th hydroxylation, that is the inactivation is taking place in the liver. So you will end up with 24 hydroxy calcidiol. Okay, now let's have a look at the name so that you're not confused. Cholecalciferol, that is only having one hydroxyl, it is known as calciol. Then after 25th hydroxylation, 2 hydroxyls are there, so that is known as calcidiol, that is 25 hydroxy cholecalciferol. Now after the third hydroxylation in the kidney, that is at the first position, you end up with calcitriol. And what is calcitetrol? When you have 4 hydroxyls, okay, one at the first position, one at the 24th, one at the 25th, and one which was originally present in calciol. So calcitetrol is your 24 hydroxylation of calcitriol. Okay, now let's talk about the functions of vitamin D. So it is involved in regulation of calcium and phosphorus levels in the body. Therefore, it is very important in bone and teeth development. Then it has immunomodulatory functions, anti-proliferative roles. During bone development, vitamin D helps in bone mineralization. Now, talking about the regulation of calcium and phosphorus. So 3 main factors are there which are involved in maintaining the calcium levels in your body. What are they? Your vitamin D, that is its active form calcitriol, your parathyroid hormone, and calcitonin. So these are the 3 factors which maintain calcium level in the body. And what are the sites where your calcium regulation is taking place? It is taking place in the intestine, kidney, and the bones. So these 3 factors, they are going to act as these 3 sites, that is intestine, kidney, and bone, and they're going to regulate your calcium levels in the body. Now, as a result of regulation, what is going to happen? Your calcitriol, that is your vitamin D, it increases serum calcium and it increases serum phosphorus. Whereas your parathyroid hormone, it increases serum calcium, but it decreases serum phosphorus. Whereas your calcitonin, it decreases serum calcium. Calcitonin, you can remember tonin. Okay, so it is tonic for the bones. So all the calcium is going to be deposited in the bone. So it is going to decrease serum calcium because all the calcium is going to be seen in the bones. So serum calcium is going to decrease. So remember it like that. It is tonic for bones. This is just a way to remember, okay? Now, action of vitamin D and parathyroid hormone in the intestine. So your parathyroid hormones favors alpha-1 hydroxylase. You already discussed that at low phosphate levels in parathyroid hormone, they are favoring alpha-1 hydroxylase in the kidney. Now, if alpha-1 hydroxylase is activated, it increases the level of active vitamin D, that's it, calcitriol. Okay. So your parathyroid, the action of vitamin D and parathyroid hormone is same in the intestine. Now what happens? Your calcitriol level is increased, which increases the level of a particular calcium channel and a calcium binding protein, which increases the absorption of calcium from the intestine. So basically what is happening? Your parathyroid hormone is going to activate your alpha-1 hydroxylase, and because of which your active levels of vitamin D, that is calcitriol, is going to be increased. Now this calcitriol, it is increasing a particular transport channel and a calcium binding protein in the intestine. In short, your vitamin D and parathyroid hormones, they increase absorption of calcium from the intestine. So just remember that your vitamin D and parathyroid hormone, they increase absorption of calcium from intestine. Okay, and your vitamin D in the kidneys, it increases reabsorption of calcium and phosphorus from the distal tubules. Okay, so in the distal tubules, they're going to increase the expression of, again, calbindin and your calcium transport channel, different calbindin and calcium transport channel, different than the one in the intestine. But what is the result of that? That your reabsorption of calcium and phosphorus from the distal tubules is going to be increased. And the action of parathyroid hormone in the kidneys, it favors calcium reabsorption. So your parathyroid hormone is going to increase calcium reabsorption, but it causes phosphorus excretion. Therefore, parathyroid hormone increases serum calcium and decreases serum phosphorus. Now let's have a look at the action of vitamin D and parathyroid hormones in the bones. So your osteoblast, they have a rank ligand. What is osteoblast? It is the bone forming cells. Okay, so your osteoblasts, they're involved in bone formation. Okay, so your osteoblasts, they have a rank ligand. The receptor for the rank ligand is in pro-osteoclast. What is osteoclast? They are involved in bone resorption. Okay, so your vitamin D and parathyroid hormones increases the action of rank ligand. This favors the conversion of pro-osteoclast into mature osteoclast. Now, mature osteoclast binds to bone and leads to bone resorption. So, because of this, what is going to happen? Vitamin D and parathyroid hormones, they increase calcium by increasing osteoclast activity, which causes bone resorption. So, if I try to explain this to you, this is what you can consider this as osteoblast. Okay, osteoblasts are what? They're involved in bone formation. Now, these osteoblasts, they are having a ligand, this known as rank ligand. And the receptor for this ligand, this is the receptor. This is present where? In pro-osteoclast. Pro-osteoclast. And what is pro-osteoclast? It is involved in bone resorption. Okay, this is the receptor for rank ligand. So your parathyroid hormones and vitamin D, it is going to increase the activity of this rank ligand, as a result of which your pro-osteoclast, it is converted into mature osteoclast. And this mature osteoclast, it binds to bone and increase the osteoclast activity by converting the pro-osteoclast into mature osteoclast. This mature osteoclast binds to bones and increase bone resorption. Okay. And by increasing bone resorption, what is going to happen? They increase the serum calcium. Okay. Now, action of calcitonin in the bones, it decreases serum calcium by increasing osteoblast activity. So this is just the opposite. So all the calcium is going to be seen inside the bone. So your serum calcium is going to decrease. And this is because of the action of calcitonin. Now, talking about the immunomodulatory action of vitamin D. So your antibacterial action of vitamin D can be seen in prevention of Mycobacterium tuberculosis. So apart from the kidneys, conversion of calcidiol to calcitriol is also taking place in the macrophages by cytochrome 27b. So level of calcitriol increases inside macrophages. When Mycobacterium tuberculosis binds to toll-like receptors on macrophages, it increases the activity of cytochrome 27b, which in turn converts calcidiol into calcitriol. And these calcitriols inside the macrophages, they favor the formation of cathelicidin, which is an antimicrobial peptide from the defensin family, thus kills the bacteria. So again, I can explain this to you in an easier form with the help of a diagram. Okay. So let's take this to be as a macrophage. Okay. This is a macrophage. And apart from the kidneys, it says that in the kidneys, what was happening? Your calcidiol was changing into calcitriol, and calcitriol was the active form. But apart from the kidneys, this calcidiol changing into calcitriol, this is your calcidiol changing into calcitriol. It is also taking place in the macrophages when with the help of cytochrome 27b. Okay, so your calcidiol is changing into 125 dihydroxy calciol, or this is calcitriol. Okay, so this calcidiol changing into calcitriol is taking place in the macrophages as well by cytochrome 27b. Now what happens? This cytochrome 27b, this is activated when it is activated by your toll-like receptors. So macrophages, they have these toll-like receptors, and when to these toll-like receptors, your Mycobacterium tuberculosis. Okay, these are your toll-like receptors. And to these, when Mycobacterium tuberculosis comes and binds, so these toll-like receptors, they are going to activate your cytochrome 27b, which is going to activate the conversion of calcidiol into calcitriol. Now, this calcitriol that is formed, this activates the production of cathelicidin, which is an antimicrobial peptide, and it is going to kill Mycobacterium tuberculosis. Okay, so this is how your vitamin D is involved in immunomodulatory action. Now, talking about the vitamin D deficiency. So it causes deficiency of vitamin D is going to cause demineralization of bones, leading to rickets in children. Very important, very frequently asked, and osteomalacia in adults. So talking about the clinical features of rickets, it includes bone deformities. That is, your bones become soft and pliable. Weight-bearing bones are bent. There's bow legs, knock knees, ricti rosary. That is, what is ricti rosary? You have nodules in costochondral junction. Costochondral junction. Then you have single frontal bone, pigeon chest, Harrison sulcus, or Harrison groove. That is softening of the ribs. So you can see bow legs like this and knock knees. Your knees are together and legs are apart. That is knock knees. This is your ricti rosary and Harrison sulcus. You can see nodules in the costochondral junction. This is your Harrison sulcus. Then talking about the types of rickets. So We're discussing the two important types here, that is nutritional and renal, although there are other types as well. So, nutritional rickets, there's insufficient exposure to sunlight or deficiencies in vitamin D consumption occur predominantly in infants or the elderly. Vitamin D deficiency is more common in northern latitudes. We're talking about renal rickets, it is in case of kidney diseases, okay? If vitamin D is available, calcitriol is not synthesized. So if you're having any renal defect, your active form is not being synthesized. It responds to administration to calcitriol. We're talking about the clinical features of osteomalacia. That is, when you have vitamin D deficiency in adults, it leads to osteomalacia. You have soft bones due to decreased mineralization and more prone to fractures. Talking about the toxicity of vitamin D, excess vitamin D gets deposited in soft tissues and blood vessels known as calcinosis. It causes contraction of blood vessels leading to hypertension. Toxicity is seen only with supplements. So apart from that, if you're not taking supplements, toxicity as such is very rare. Excess vitamin D produced in the skin is converted to inactive form. Now talking about the RDA, that is required daily allowance. In children, it is 10 micrograms per day. In adults, it is 5 to 10 micrograms per day. And in pregnancy, it is 10 micrograms per day. So we've taken the important vitamins, that is A and D. Hopefully in the next turn, we'll finish the remaining of the vitamins. The important ones we've taken them in a little detail because they're very important and very frequently asked. So if you have any queries, you can ask. Your rickets is very important. And your vitamin A is also very important vitamin. Okay, its synthesis, metabolism, and everything. So with this, we come to the end of the lecture. See you in the next class. Thank you for watching.
डिस्कनेक्ट कर दो सब नेट डिस्कनेक्ट करो